Cats are liquid. They can flow, they have a fixed volume, and they take the shape of their container.
By that logic, your cells are also liquid. Even the molecules in your cell membrane can move past each other. Okay, but Campbell's clearly not a liquid. Yeah, they're not. The best term to describe your cell membrane would be fluid, not liquid. Fun fact, the fluidity of cell membranes depends on the situation. The cell membrane needs to be stable enough to hold the cell together, but fluid enough to allow molecules to pass through. Fluidity depends on 3 factors: temperature, type of phospholipids, and cholesterol.
Cholesterol? Wasn't that bad for you?
I'll explain each one. First, temperature. The higher the temperature, the more phospholipids move around. Second, phospholipid structure. Long, straight fatty acids pack tightly, whilst the opposite is true for short, bent fatty acids.
Wait, I still don't get how being long helps.
Fatty acids are weakly attracted to each other. The longer the fatty acids are, the more surface area to stick to each other.
Oh.
Third, cholesterol. Does it increase or decrease the fluidity? It depends. Cholesterol is weakly attracted to the fatty acids. At high temperatures, which is when membrane fluidity
increases,
cholesterol acts like a glue to keep the fatty acids together, decreasing membrane fluidity. At low temperatures, where membrane fluidity decreases, cholesterol keeps fatty acids apart, increasing membrane fluidity.
Wow.
Membranes are also fluid because they're flexible. They can pinch off to form vesicles. You can use this for exocytosis, which is when substances are transported out, and endocytosis, the opposite. So, are you sure Campbell cells aren't just liquid? Very sure. Otherwise, how would their cells hold together?
Are they even held together?
Yes, by a special type of molecule called cell adhesion molecules, or CAMs. Oh, CAM for Campbell!
H2O, you said the membranes can be pinched off for transport, but eventually wouldn't that leave us with no membrane?
Good question. The vesicles are smaller than they usually appear on diagrams, so they don't actually use that much membrane material. Also, membrane material is constantly recycled. Another reason is that these are not the only methods of transport. Molecules can also travel by diffusion, active transport, through channels, through cotransporters. Whoa, slow down. I'll explain the most interesting ones. The sodium-potassium pump, gated ion channels, and glucose transporters.
They seem hard to understand.
Not when you understand them.
Well, yeah, duh.
Sodium-potassium pumps use ATP to pump 2 potassium ions one way and 3 sodium ions another way. What's the point of doing that? You need them indirectly to think and to absorb nutrients.
How do ions help with thinking?
Neurons relay information using changes in membrane potential, which is the difference in charge across the membrane. Sodium-potassium pumps help neurons establish the baseline membrane potential.
Oh.
When a neuron is stimulated, ion channels open, which change the membrane potential.
Oh.
A more detailed explanation is in the video on neural signaling. Here, the sodium channel was stimulated by a voltage, so this is a voltage-gated ion channel. There are also ligand-gated ion channels, which are opened after a molecule binds to them.
What about absorbing nutrients? I'm more interested in that.
Because you like to eat?
Of course.
In your small intestine, glucose is absorbed using sodium-dependent glucose transporters. Glucose cannot cross the channel alone, so sodium must cross with it. Good news, there is a lot of sodium outside the cell because there are sodium-potassium pumps nearby.
Why did Mother Nature have to complicate things?
It didn't mean to. Molecules move from an area of high concentration to low concentration. For glucose, that doesn't always mean moving into the blood, so it needs an extra push from sodium. It's just like how a stronger person can help a weaker person go through a revolving door.
I see.